right, so now we're recording. So yeah, most of the questions on that final exam study guide are just, basic review of the knowledge that we've covered so far this term. There are a few questions that seem weirdly specific on there. Just note that that's there for a reason. So keep those in mind, because there's some questions on these exams that I just don't fully agree with. So those are my hints to you. So let's take a look at it. Let me pull it up so that we can all kind of see it together and talk about it. Okay. All right, so there's the final exam study guide. All right, so a lot of, just basic terminology here. So just being familiar with those terms is going to be important to be able to answer the questions on the final. And then we start to get into questions about what we've learned so far this term. So I think maybe I'll let you guys drive the bus here and tell me if there's any questions here that you don't feel you understand or you need my help answering, or if you've got just general questions about the final.
I had a quick question about question 36. My thought was that you would figure it out through a titer, but that's not through looking at it. That's like an actual experiment. So I wasn't sure, because the question was by just looking at the test tube, how would I know how many bacterial cells are present?
Okay, yeah, so that's a good question. So basically what you can do by just looking at a tube is examine turbidity, right? So turbidity is the key word there. And that's like the cloudiness of the solution. So if it's clear, that means there's no cells present. If it's a little cloudy, there's a few. If it's super cloudy, that's a lot.
Okay.
Okay. I lost my chat window, so if anyone's popping up in the chat, I don't know where I put you. Sorry. There it is. Okay. All right. Any other questions that we ran into going through this study guide? I'll throw some at you. All right, so what is peptidoglycan?
A polymer consisting of sugars and amino acids that form, let's see if I can read my own handwriting, to form a mesh-like layer outside the plasma membrane of most bacteria, consisting of that N-acetylglucosamine and N-acetylmuramic, and then it confers the mechanical rigidity of that cell. So that's the structure.
Perfect. Yep, perfect. So yeah, it's essentially the cell wall of the bacteria, but it's made of all of those little components, and it helps protect them and maintain their shape, and so that they don't lose their osmotic balance. What about lipopolysaccharide? What's that?
The outside membrane. Must not have answered that one.
Okay.
That's the outside of gram-negative bacteria.
Perfect. Yep. Yeah. So that's the outer membrane present in gram-negative bacteria. It's also known as an endotoxin, right? Because it causes systemic response in its host. All right, let's think back to a little bit of our cellular synthetic processes. So if I'm copying a DNA molecule, that's replication. If I am taking part of that DNA molecule in order to create a protein from it, what's that process called?
Translation?
Yeah, so translation is the generation of the protein. What's the process? I should have been more clear in my question. Where we go from DNA to RNA, making the RNA.
Oh, transcription.
Mhm. Good, transcription. So when we go to finish the transcription process and we have to cut out some of the DNA that doesn't belong and keep the good segments of DNA, what are those good segments of DNA called?
With expressing like X.
Good. Yeah, good. Exons.
Exons.
Excellent. Yeah. Very good. Okay. How do bacteria replicate?
Binary fission.
Very good. Binary fission.
Was the AZT a specific question that you were like, because I don't remember reading about that too much.
Yep. Yeah, very good. So yeah, AZT is it's an antiviral drug that's used to treat HIV.
Yeah, I had to look that one up.
Yeah, so that one popped in there. Just, yeah, that's the kind of question. Like if you see something like that, there's a reason I put it on that study guide so specifically.
Okay.
Okay, all right, we understand our bacterial growth curve, the 4 different phases of it. So when we initially plate bacteria on a petri dish, we'll have that initial lag phase, and that's while they're adjusting their metabolism to their new environment. The logarithmic growth phase is when they're just growing like crazy, exponential growth. A stationary phase is when new births, in quotation marks, equal deaths. So we're kind of at a stationary, no change. And then the death phase, why does it say dead? The death phase is when they're dying more than they're being born. Okay. What are our 2 major types of fungi?
Yeasts and molds.
Very good. So yeasts are single-celled fungi, molds are multicellular. The cell wall of most fungi is made of
Chitin.
chitin. Very good. Okay. All right, so thinking back to our antimicrobial drugs, what's the main target of penicillin?
Peptidoglycan.
Yeah, right, very good. So penicillin targets the cell wall, so it prevents bacteria from basically creating more peptidoglycan, so that as their old peptidoglycan wears out, they tend to break apart. So given that it works on peptidoglycan, do we think it's more effective against gram positives or gram negatives?
Gram positive.
Very good. Okay, getting into the immunity section, because I know a lot of people have problems with this. And immunity is one of those things that when we first cover it in micro, yeah, Kira, you are on a roll. When we first cover immunity in micro, it can be confusing for a lot of people. It's usually one of the, especially when we get into specific adaptive immunity, it's one of the more difficult concepts that we cover. We're going to cover it again. So we'll cover the immune system all over again when we get into A and P. So this is one of those things that if it doesn't quite all fit into place just yet, that's okay. And we're going to add more detail to it and make it make more sense as we move forward in your course progression here. So So talking about adaptive immunity, so your specific immune system, I guess I kind of gave the answer there. What are the major types of cells that are involved in adaptive immunity?
B cells and T cells.
Yeah, B cells and T cells. And both of those cell types are going to be made in the bone marrow, just like all blood cell types are made in the bone marrow. And then they migrate to different places to mature. So the B cells are going to mature, they'll stay in the bone marrow. The T cells will mature to your thymus gland. Now, one of those two cell types creates antibodies. Which one?
B.
Yeah, B cells create antibodies. So we've got 5 different types of antibodies. So those are, well, I'll tell you the type, you tell me what they do. All right, so the IgG, what's IgG all about?
Does anyone else want to go?
That antibody is produced second and is capable of binding complement, and I can't read my writing, and can cross the placenta.
Very good. Yes. Yeah. So that's yeah, you're right. It's usually kind of lags behind the production of another antibody in an initial infection, but it is the most numerous antibody that you have circulating at any given time. It is the only one that crosses the placenta. So very good. What about IgM?
That'll be your first one present, and it contributes to like the activation of the B cells.
Very good. Yeah. So IgM is that the first one that you produce in mass quantities upon an initial infection, and so that's going to be the important kind of initial wave of antibodies that helps you start to get better. What about IgA?
It's produced after the infection has cleared and is expressed in saliva and breast milk to protect against future infection.
Perfect. Yep. So IgA is the one that we find in secretions. Yeah, so mucus, saliva, breast milk. And that one is going to be, yeah, important for future infections. Anything that's going to be trying to cross those mucous membranes, the IgA is going to be there to help protect. What about IgD?
That's your second. That comes after the IgM, and the function is to signal the B cells to become activated. Like when they make like the antibodies or something.
Yeah, that was kind of a trick question. They don't really, we don't know. Yes, that's what they do, but in a similar way that's what all antibodies do. But yeah, we don't know. What about IgE?
Immunity against parasites and is active in the immune system for allergens.
Yes, very good. So allergies and worms, essentially. Okay, so that's good, the 5 types of antibodies. All right, so you should be familiar with all your different types of blood cells. Know the difference between anabolism and catabolism? Which one's building cells up or building molecules up?
Anabolism.
Very good. Yeah, anabolism builds up, catabolism breaks down. All right. What's competitive inhibition?
Those are the ones that bind the active sites of those enzymes that prevent the actual substrate from making that molecule.
Perfect. Yeah. So this is a competitive inhibition is a way that we can control the activity of enzymes. So we can inhibit certain enzymes competitively by creating a molecular mimic, and that mimic will bind to the active site, preventing the enzyme from catalyzing any future reactions with the actual substrate. Very good.
Are allosteric ones, are those part of competitive inhibition, or is that something separate?
That's a different kind of inhibition. So the allosteric ones, instead of binding directly to like the active site of the enzyme, they bind to a regulatory site on the other side usually. And when they bind to that regulatory site, it causes a change in the shape of the active site so that it can no longer catalyze reactions. But it doesn't directly compete with the substrate. It just changes the shape of it, of the active site of the enzyme. Good question.
Do you want to answer 51, how the yeasts and molds reproduce?
Oh, sure. So yeasts reproduce by a process. Well, there's a couple ways that yeast can reproduce. So they can reproduce by budding. And so budding is a process that looks a lot like mitosis, right? So mitosis is when we kind of double ourselves and then split in half evenly. Yeasts can do budding, and instead of like an even split, it's like you'll have this parent cell, and then you'll get a little bitty bud growing off of that parent cell, and that little bitty bud will grow bigger and bigger and bigger, and then they'll split. So they can reproduce by budding. They also reproduce by a process called Pseudo Hyphae. Pseudo, I'll type that in the chat. All right, so budding or Pseudo Hyphae. Pseudo Hyphae look like, they look a lot like buds, but they're just more elongated buds. Molds reproduce by the production of spores. So they, the spores, are present on the end of these stalks, so long little stalks that are called hyphae. At the end of the hyphae are these spores, and those spores then go airborne, and that's how molds reproduce. So it's too bad we didn't get to be in the lab together, because if you look at a mold, you know, when you say your bread gets moldy or whatever, and it looks kind of fuzzy, if you look at those structures underneath the microscope, they're basically these tiny little hairs, these hyphae, with little spores on the end of them, and that's what gives it that kind of fuzzy looking appearance. Right, so that's how molds reproduce. Okay. Yeah, so good question, and definitely just stop me if I'm passing by important ones. I'm just sort of picking randomly here. Good morning, Angel. Okay. What's a naked virus?
A virus containing only the nucleic acid and its capsid. It doesn't have that envelope.
Perfect. Yeah. So just nucleic acid and capsid, no envelope. If a virus is enveloped, where does that envelope come from?
The host cell when it is endocyte or exocytosis.
Perfect. Yep. Yeah. So it's basically created from stolen phospholipids from that host. Okay. Yeah. Okay, so, and we understand how we create RNA from DNA, the pairing of the different nucleotides. We talked about transcription and translation. So transcription is DNA to RNA, translation is RNA to protein or polypeptide, however you want to phrase it. Okay, the steps of cellular respiration. So this is another one of those concepts that everybody hates, and we'll cover it now. We'll cover it again in A&P, just because it's incredibly important for staying alive. So we just need to understand how it works on a at least a simple level, understand the basics of it. So your 4 steps of cellular respiration are glycolysis, then we've got preparatory reactions, the Krebs cycle, and the electron transport chain. So you should be familiar with those 4 steps there, and then the basics of what's going in and what's going out. We don't need details, just a simple idea of what's going in and what's going out and where each of those steps is happening inside the cell, right? So like, for example, glycolysis is happening in the cytoplasm, right? And in order to kickstart glycolysis, we need one glucose molecule. We're gonna need to pay a little bit of ATP to get us going. So 2 ATPs go in, right? And so that's gonna help us break that glucose molecule down into 2 pyruvates. And then we're also going to load up some, she's making fun of my hand motions. We're gonna load up some electron transport, or load up some electron carriers. Right, so and that's what the NAD, NADH, FAD, FADH2, those are electron carriers. They are coenzymes. So a coenzyme is an organic molecule that's attached to an enzyme, and that helps that enzyme carry out its reaction. Right, so in this particular case, these guys are just there to hold on to high energy electrons for us. So NAD is without the electrons, NADH is when it is carrying the electrons, same for the FAD. Yeah, so we're loading up those electron carriers really in all steps. So we're loading up electron carriers in glycolysis, in the prep reactions, in the Krebs cycle. How about a better question? What are they made from? So what do we make these guys from, these coenzymes?
Are you looking for like the specific name, like
in your diet? What are you eating in your diet that you need in order to create these? What's that?
Are you looking for glucose?
No, not glucose. Just for these specific coenzymes here.
Pyruvates?
They're actually all made from, they're derived from B vitamins. So this is why you need B vitamins in your diet. It's one of the reasons that you take B vitamins to, to help your energy and metabolism. Yeah, very good, Justina, B vitamins. Yeah, so because you need the B vitamins in order to manufacture these coenzymes. All right, and so wait, let's clear up what we're talking about here. So cellular respiration is the breaking down of that glucose. What's the final product? What's the point?
ATP.
ATP, good, yeah. So the whole point of that cellular respiration is to get ATP. If we're performing cellular respiration efficiently, we're gonna be using oxygen. Right, so that's why we need mitochondria to do the rest of these steps here. So prep reactions, Krebs cycle, and electron transport chain. We need the mitochondria, and mitochondria uses oxygen. You have some cells in your body that do not have mitochondria. So in those cells, what are we doing to produce energy? What's the process if we don't have oxygen?
Fermentation.
Good, yeah, so we're gonna do fermentation. And that can also happen, especially in skeletal muscle cells. So cells that are really, really high energy cells that are going through a lot of oxygen. If they run out of oxygen, then they can switch over into the lactic acid fermentation. right? But the whole idea is to, and in fact, the entire reason why you need to breathe in oxygen is to more efficiently turn glucose into ATP. That's the whole point. Okay. All right, what are some virulence factors?
Adherence factors like pili and fimbriae that make them adhere to cells.
Good. Yeah, so anything that that is going to help promote attachment. So fimbriae, pili, capsules are considered virulence factors because they're sticky and they kind of increase the size of the bacteria, making it hard for white cells to phagocytose the bacteria. Things like toxin production, so either endotoxin like lipopolysaccharide is considered a virulence factor, or exotoxins, which are, so exotoxins are compounds that are produced that instead of like an endotoxin where it basically causes this systemic response like fever and just all kinds of malaise and nasty feelings, an exotoxin is a compound that's going to attack a particular type of cell. So for example, some staph produce exotoxins called hemolysins. My 13-pound dog is trying to destroy the 80-pound dog that's walking out front. You're not going to do it, buddy. So hemolysins are, there's just a weird scene happening out my front now. The hemolysins are particular compounds that destroy red blood cells specifically. So that's what an exotoxin is. It attacks a really specific type of cell rather than having this whole body effect. Oh. Really sorry about that. This is Jack. Jack just had a little seizure. He sometimes gets a little excited and loses control. Poor baby. We're going to the vet. We've got an appointment next week, so sorry about that. He gets so excited and then he just freaks out and then loses entire control, and hopefully he doesn't pee on me right now because that's usually the next step in the process. Okay. What was I talking about? Exotoxins. So yeah, again, they're just specific compounds that are considered virulence factors. So any kind of enzyme production that is considered toxic, actual toxins, adherence mechanisms, all of those things are considered virulence factors. Okay. What causes amoebic dysentery?
Is that the Entamoeba histolytica? Very
good. Yeah, so amoebic dysentery caused by Entamoeba histolytica. So that's a, what category of microbe does an amoeba fall into?
Protozoa.
Good. Yeah, protozoa. I don't remember if this question is on there, but we should, I don't think we mentioned it in class. So primary amoebic meningoencephalitis. Did anyone look up what causes that?
I don't know if I can pronounce it, but it's that Naegleria fowleri.
Good. Yeah, Naegleria fowleri. This is a really weird one. I'm sad that we didn't get to be in class because then we could talk about more of this stuff. So Naegleria is an amoeba that's like just a normal soil bacteria. He lives in the dirt, and he's totally free living. So this is not a parasite per se. But if he accidentally finds his way in your nose, so say you're swimming in a lake or a river where there's like sediment, there's like dirt in the water, and you get that water up your nose. So if he finds his way into your nasal cavity, he will follow your olfactory nerve, your sense of smell nerve, into your brain, and then basically eat your brain from the inside out. He turns your brain into Swiss cheese within like 2 weeks. It's a terrifying disease. That's really rare because you have to get this one weird amoeba right up into your nasal cavity. But it does happen more often in the South. So that particular amoeba doesn't deal with freezing temperatures too well, so we don't have them really around here. But it's one reason to just be careful not to get water up your nose if you're going swimming in a lake or a river down south. The other common, so not common because it's rare to begin with, but another way to get infected is through the use of neti pots. So those pots that you pour the water up into your nasal cavity. So always a good idea to use bottled water when you use one of those and not from your tap. All right, so do I need a HEPA filter if I'm in a BSL-4 laboratory? Yeah, and then some, right? We need a lot of protection if we're in BSL-4. How do bacteria share genetic information?
Horizontal gene transfer.
Very good. Yep, so horizontal gene transfer, since they reproduce by binary fission, they're essentially just making copies of themselves, so they don't have the benefit of sexual reproduction in order to switch genes around. So they'll use horizontal gene transfer. And that there are 3 different types of horizontal gene transfer. What are those?
Transformation, transduction, and that conjugation.
Very good. Yeah. So conjugation is the one that is a direct transfer by the formation of a pilus. So one bacterium that has a plasmid in it, and that plasmid has genes on it for the creation of a sex pilus, will create that pilus, reach out, grab another bacterium, and then send a copy of that plasmid over to that other bacterium. And so conjugation is one of those things that it's cool. It's a way that they can get their genetic material all switched around. It's also dangerous in that it's a way that they can transfer genes for antibiotic resistance. And it doesn't even have to be the same species. So we can have species that are good, like positive, normal microbiome species, can transfer genes for antibiotic resistance to, can transfer genes to pathogens, right? So that's not great. All right, so that's conjugation. Transformation is not a direct way of horizontal gene transfer. This is when the donor, the donor DNA, that cell is dead, and the DNA is just kind of floating around in the environment. If a cell is competent, meaning it's got DNA binding proteins on its surface, it can pick up that kind of floating DNA from the environment and bring it on board. And then the last one, transduction, that's when we transfer the DNA indirectly through a virus. So a bacteriophage attacks one bacteria, and usually that bacteriophage is going to destroy that bacteria through the lytic cycle. but not before it uses the bacterium to make copies of itself. So, and as it's making copies of itself, every so often it brings some of the bacteria's DNA along with it, and it can use that to inject that DNA into another bacterium. How many questions on the final? There are 50 questions. Right, so it'll be just, should be similar to the midterm, although hopefully not the same issue with repeat questions. I really didn't like that. I made my opinion known, so hopefully we won't have that problem again. Okay. Yeah, those repeat questions, like what was the point? It doesn't make any sense, because if we want to be testing your knowledge, you know, just pounding the same question over and over and over again is not really giving us the breadth of knowledge that you actually have. Is it on Monday or Wednesday? So yeah, your final exam is going to be Monday morning at 9 a.m. If you cannot make it to that 9 a.m. session, then we'll have a second opportunity at 4:30, just like we've been doing the whole time here. So either 9 a.m. or 4:30. It will be 50 questions. You'll have 90 minutes to take it. So, and you probably won't use that entire time.
If there are repeat questions, I told everybody before, don't answer it the same way if you don't know the answer.
Spread your risk.
Well, you get a 50-50 shot at getting it right then. I mean, if you don't answer it the same way, because you're going to get them both wrong then.
That's true. Yeah. So unless you're sure, if you're sure,
yes,
answer the right answer every time. I don't think that that's going to happen again. That was poor formatting. And it makes you second guess yourself. And second guessing is one of those issues that really is problematic for a lot of students, right? So you're taking a test, you feel pretty confident, you've been studying going in, and then you read a question and you're like, is it this or is it that? You have kind of like a first hunch of what you think your answer is. And then you overthink it, and you keep thinking about other ways to interpret the question, and you change your answer. That's usually a recipe for disaster. And I know that it's hard not to do it, but unless you're sure that you read the question wrong the first time, go with your gut. Go with that initial hunch of what your right answer is. Yeah, because once you start overthinking, you will confuse yourself, right? And you'll get into a mind space where you're no longer using kind of your logic and your knowledge to answer the questions. You're getting a little bit emotional because you're worried, and that's not where you want to be when you're taking a test, right? So we want to get out of that emotional brain and into your logic brain. Yeah. So I know it's easy for me to say, hey, don't be nervous. Don't overthink it, but seriously, try to do your best to kind of go with your gut. Yeah, hard doing it at home. So that's another thing. If you can, I mean, you've probably been doing this already, but trying to, get yourself a quiet space where you're not going to be interrupted, where you can sort of focus on the test and not worry about what else is going on. Like me, for example, right now, like if I were taking a test right now and the dog ran by the front window and that caused my dog to have a seizure, and like, this is not a good test taking environment. So try to do your best not to follow my lead here. Is it worth 100 points? Caroline asked. Yes. Yeah. So this is 100 points, meaning it's worth 10% of your grade. All right. Any other questions about the final or anything else as we sort of wrap up the term here?
When should we have these forms turned in? By tonight or by Sunday?
By Sunday.
Okay.
Yeah. So take your time. Yeah, because use this to study the material, and just get them in to me by Sunday night.
How are we turning them in?
You can just submit the file as an attachment into the Unit 8 discussion.
Okay.
Oh my God, you got a neighbor coming to tell you your daughter is outside using some sunscreen up. Oh my gosh, see, it's that kind of, it's that kind of stuff that like, it's really challenging to work from home. And I feel you guys, I understand like with all of the interruptions that we have in our home lives, especially if you have kids. I just saw my kid come climb on my lap. Like it's not easy. So you guys should be proud of yourselves that you're getting this done in spite of all of these obstacles that are being thrown at us from Corona amidst other things. So, almost there. Any other questions, you guys? All right. So I'm going to go ahead and then, yeah, you should be pros for your next courses. So yeah, by the time, so most of you, I imagine, are progressing to A&P 1 next term if you haven't already taken it elsewhere. That's going to be so much better. One, it'll be better for me because it's the only course I'm teaching next term, so I'll be able to focus more and get your grading in on time. And it should be better for you guys as well, just because it's more focused on the type of material. I think it's, I'm going to stop recording. Hang on.